Well, everyone, hello and welcome. We are on to chapter 11, which is going to be all about cell division. And you might be asking, why do cells divide? Okay, well, first of all, let's just kind of go back to the basics here. Is we are multicellular organisms comprised of around 60 trillion cells. That's trillion with the T. Now, some of those cells will never divide, as in they are made, you're born with them, or they're developed later in your pre-adolescence. They're made, that's it. If something happens to them and they die, then there's really nothing that can replace them outside of some seriously advanced biotechnology or medical techniques. However, there's some cells that constantly divide, meaning that they are actively mitotic. These are cells that have a finite lifespan, like, for example, cells called keratinocytes, which make up your skin. In fact, basically everything that you see on the surface of your body is dead because the skin cells, the keratinocytes that are on the surface of your skin, they've actually been killed a while back and just filled with the tough protein called keratin. So there's really few living cells that you see on the surface of your body. And if you do see any living cells, that's bad news because that means your internal environment is significantly exposed, which you do not want. So yeah, it's kind of funny. There's very little of our body that we can see that's actually comprised of living cells. But anyways, cell division occurs for basically more or less four reasons. And I'll also give you some examples as we go along here. So cell division is the process of existing cells undergoing a process where they create copies. Now, in the case of mitosis, these are genetically identical copies, whereas with meiosis, they are genetically unique. Genetically identical. And in the case of meiosis, which we'll be going into more detail later on, in the case of meiosis, they are genetically unique. Also, a note here is some students get confused because many sources, they actually split up, and this is just a note for when you get to it. They actually split up prophase into two phases, prophase and prometaphase. I don't do that for ease of teaching because I'm trying to teach a concept here, not memorization, but a concept. For ease of teaching, I keep prophase and metaphase two distinct stages. I do that because, as I said, I'm trying to teach the concept, not trying to teach memorization, but the concept. And I think it's easier to teach the concept if you make prophase and metaphase two distinct stages. So something just to keep in mind as you watch the rest of this lecture. But anyways, most mitosis happens with something called stem cells. And stem cells are cells that can differentiate, meaning that they can differentiate into other types of cells. So think of a stem cell as like a baby. That baby can grow up to become anything it wants to. But when it's born, it's basically a blank slate. That's kind of what stem cells are. Is that when they are is that you have an existing layer of stem cells that constantly divide, and then cellular signals they tell that cell what to become. For example, on the bottom of your skin, the very lowest layer of the epidermis, there's a layer of constantly dividing stem cells, and they turn into skin as they mature. They're actually given signals by other skin cells telling them to become skin cells. also known as keratinocytes. And stem cells are found in areas where the cells must be constantly replaced. Skin is a great example because your skin is actually designed to be damaged. Of course, in today's developed world, having damaged skin does not really seem like an appealing thing. But the idea of the skin, the reason why it's so good that the cells are dead is that you can't kill what's already dead. So let's say that you spill something on your arm, even if it is slightly corrosive, it's not going to damage the living parts of your body. If you get a cut, no big deal, the cut will heal over a period of time. So the skin is a great example of something that is, or an organ that is constantly mitotic, because the skin cells are designed to be replaced. Similarly, in cells that line surfaces of cavities, the cells that make up your digestive system, the digestive system, they're constantly turning over because you need those cells to operate at peak efficiency, and older cells don't work as well. So those cells, the cells that line the small intestines, they actually die about once every 7 days and replaced with new ones just to make sure that they're operating at peak efficiency. Other examples are erythrocytes, also known as red blood cells. And they actually, red blood cells actually have a very finite lifespan because since they're literally just bags of a protein called hemoglobin, they actually don't have the ability to sustain life or their own life. So after a period of time, those red blood cells are actually broken down by the liver and turned into a compound called bile. And then there are stem cells in your bone marrow that keep on producing new red blood cells. So anyways, there are certain components of cell division, and one is cell signals. We'll be talking about these briefly. But what cell signals do is that they tell the cell when to begin the process of mitosis. So keep in mind, mitosis is not just a snap of your fingers and here we go kind of thing. Okay, well, backtrack. In bacteria, that is kind of the case. It's more of a process of preparation that once this process begins, the cell either has to undergo mitosis or die. That is those are the rules. 2, you must have DNA replication. What this means is that the existing copy of DNA in the cell must be copied. So there must be an exact copy of DNA synthesized. DNA is not just copied, it actually has to be synthesized or made anew. And what happens is that you have enzymes in the nucleus that actually will read the DNA, and they'll actually use that thing that you read about in chapter 4. They'll use the concept of complementary base pairs to build an existing or a new exact copy of that DNA molecule. Because the point is, of course, is that one cell that's undergoing mitosis, that one cell needs to divide into 2. And for that to happen, you're going to need DNA, a copy for each cell. You also need DNA segregation, and this is where chromosomes come in handy. We don't get into chromosomes too much in this course, but DNA segregation is when it condenses into chromosomes, which we'll talk about in a bit. Condenses into chromosomes and moves during mitosis. So what happens is that these chromosomes can actually be pulled two opposite ends of the dividing cell by cellular machinery. And we'll get to that more later. And we also must have cytokinesis, which is when the cells actually divide. So cytokinesis is when cell division occurs. So cells actually divide. So these are the rules of cell division. And now, this is not on the outline, but I'm going to have you add it in. We're going to talk about the process. Well, actually, it is on the outline. I'm sorry. So we are going to talk about the process now of something called binary fission or bacterial reproduction. It's called binary fission. As we talked about in lab, is that bacteria, also known as, well, they're a class of the larger category of prokaryotes, they're so simple that they divide every 20 to 25 minutes. That's how fast bacteria divide. It's also why you wash your hands very often. But anyways, in binary fission, what's going to happen actually is that the DNA is going to replicate as the cell is elongating. Now, the interesting thing about bacteria is that they have a relatively simple genome. Their genome is only a couple of million base pairs slash bases in length. So they only have actually a single chromosome. They don't have several chromosomes. They have a single chromosome. So what happens is that this cell is going to begin the process of DNA replication. And I'm going to show here that the bacteria is going to be elongating. Now, as that's happening, the DNA, so I'm not going to draw it in just a circle. What's going to happen is that the cellular machinery is going to begin to make almost like a bubble at a spot called the ori. The ori is going to be the origin of DNA replication. And what's going to happen is that this bubble is going to keep on growing. Let me go ahead and draw the bacteria that's even longer. I know, well, actually, run for your lives, everyone. It's super bacteria. And what's going to happen is that this bubble is going to keep on growing because you are having a newly synthesized chromosome being formed. So it's now going to look more like this. So notice how there's basically 2 copies of the chromosome now. Okay, I'm going to have to erase this because I'm running out of room. So now what's going to happen is that the bacteria, which is now about the size of 2 bacterium, here's your hot dog, is now going to have 2 unique sets of DNA. And it is going to divide. So cellular machinery is going to divide it, and now you are going to have 2 newly formed bacterium, each with their own copy of the chromosome. So the cell actually elongates as DNA replication occurs, which makes it incredibly efficient. Just to illustrate my point, I'm going to show you a video of binary fission in action. And of course, it's sped up, but it just shows you just how fast bacteria can divide, as I said, every 20 to 25 minutes. Okay. Okay, let me share it with you guys here. So you're seeing that just by starting with a few bacteria, look what's happening over just a short amount of time. So you're seeing now why it's so important to wash your hands once every hour or at the most couple of hours. Is you're seeing just how fast just a couple of bacteria can replicate. And at the 4-hour mark, it's basically just taken over. Okay, well, hope that was caught your interest. So that's the process of binary fission. And now, before we get to the cell cycle, let's go ahead and talk about cell cycle control. And this is something that's unfortunately broken in cases of cancer. But we're going to talk about now cell cycle control. And we're going to talk about the cell cycle in just a bit. But just to give you an illustration, I'm going to use this sort of circle to show you the different stages of the cell cycle. It's not to scale, but just to give you an idea. And each of these areas that are shaded, we'll talk about them later, represents a different stage in the cell cycle. I'm not going to tell you yet because you'll learn about it ad nauseum in the next section. And a series of events has to happen for the cell to progress through this cell cycle. And one of those biggest things is a stage or a checkpoint called the G1S checkpoint. And what this is that this is actually a point where the cell determines if mitosis is possible. It's not always. But what tells the cell to progress through these checkpoints, there's actually several checkpoints that control the cell cycle. There's actually several checkpoints, which I'll just mark here in teal, are something called cyclin CDK complexes. Now, there is a type of enzyme called a cyclin-dependent kinase. Cyclin-dependent kinase. Anything that ends with ASC is an enzyme. In case you need a brief refresher, enzymes, what they do is that they either build molecules, break down molecules, or just speed up chemical processes. So anyways, it's called a cyclin-dependent kinase because it can't work without a type of a molecule called a cyclin. Now, one thing to keep in mind is that cyclin-dependent kinases are always present. They always just hang around the cell, especially if it's actively mitotic. They're always here. But they can't do anything, and keep in mind, even though I'm saying that they're outside the cell, they're really integrated into the cell. But they can't do anything unless a molecule is made called a cyclin. And this molecule has to be produced. If it doesn't, it can't bind with the cyclin, the CDK, sorry, the cyclin dependent kinase. So when a cyclin binds with a cyclin dependent kinase, and it has to be a perfect fit, just so it isn't any molecule. When this occurs, the cell will progress to the next stage in the cell cycle. During the process of mitosis, there's actually several checkpoints where cyclin CDKs are present. But the idea behind this is that, and it's actually ingenious, is that the cell needs to be given the okay to progress through the cell cycle. And by only making cyclins when it's time for the cell to go through the cycle, it controls how often the cell divides. So even though the CDKs are constantly present, if the cell is not making cyclins, then the cell cannot go through the cell cycle. So it's a series of checks and balances. Okay, cool. So now we're going to go through the process of mitosis and meiosis. All right, over and out. Maintenance. So we have something now called a cell cycle, and it is important to note that a cell cycle is not a thing with every type of cell. So the cell cycle, it's important to note that the cycle time is variable for each cell. For some cells, it's really fast, like 24 hours. For other cells, it's measured in months or years. And in other cells, there is no cell cycle because they never, ever prepare for mitosis, as we'll talk about. So there's a couple stages of the cell cycle. And the first one is something that we call G1. So G1 lasts quite a while. G1. And G1 is the stage that occurs right after mitosis. So this is a period of normal cell functioning, meaning that the cell has been synthesized. It is now a fully functioning cell, and it's just doing its job, whatever its job is. If it's a enterocyte, it's absorbing nutrients. If it's a keratinocyte, it's being pumped full of keratin protein. If it's an erythrocyte, which is a red blood cell, it's carrying hemoglobin. So G1 is after mitosis. The cell's ready to do its job. It's ready to go. It's also very important to note that non-mitotic cells do not leave G1. So for example, neurons are incapable of undergoing mitosis. There is no mitosis for neurons. Because of that, they will never leave G1. Now, when a cell is going to prepare for cell division, it must pass through a very important checkpoint called the G1S checkpoint. This is a very important checkpoint because this is when various cellular processes determine if the cell is capable of preparing for mitosis. So right now, G1 is the cell that's doing its thing. It's doing its normal functioning. But if a cell is actively mitotic, various checkpoints in the cell, or various check systems, I should say, to not create confusion, various check systems are going to assess if the cell is capable moving into the stages of cell division. The most important checkpoint is called the G1S checkpoint. If the cell passes the G1S checkpoint, it's going to move to the next stage that we're going to talk about soon. And this stage is called S phase or synthesis. However, if it doesn't pass the G1S checkpoint, and this is very important, it's moved to almost like a cell purgatory, a side category called G0. And G0 basically means cell is told not to divide. And there's two reasons for this. One is a period of malnutrition. If there are not enough nutrients to build new cells, then obviously the cells cannot be built. So the mitotic cells are told to hold off, to not do anything. This is why malnourished children are smaller in size and stature than their nourished counterparts. The other possibility is that the cell is defective, meaning that the cell is not acting or functioning properly. And if a cell is defective, eventually it's going to undergo apoptosis, meaning it's going to be told to destroy itself, almost like if you watch science fiction shows or movies, self-destruct. So that's basically what it is the cell is told to self-destruct. Assuming the cell passes the G1S checkpoint, it's going to go into the next stage called S or synthesis. So once a cell passes the G1S checkpoint, it has to divide. If it doesn't divide, if something goes wrong and it can't divide anymore or prepare for division, then the cell will be destroyed. So once it passes the G1S checkpoint, then it is going to have to go through mitosis or be destroyed. So the S phase is DNA is replicated, meaning that you're going to have a completely new set of DNA. And of course, we'll be having an entire lecture or two dedicated to DNA replication. But basically what happens is that once you have one molecule of DNA, then you have two. It's a very complex process. Now the next one is called G2. And in the G2 phase, and this is going to be simple, but cell prepares for mitosis. So things like replicating the necessary organelles, enlarging the cell membrane to prepare to split into two. So the cell makes all the preparatory things to prepare for mitosis. And then finally, mitosis, which actually only lasts a very short time. Mitosis is, excuse me, called the M phase or mitosis. And this is the series of cell division. Okay, so now we need to talk about some chromosome terminology. So I'm going to show you here. This is what's called a karyotype. A karyotype is a snapshot of chromosomes that everyone has in their body. And chromosomes are only visible during mitosis or meiosis. What is a chromosome? Well, your DNA is actually an extremely long molecule. And DNA exists tightly coiled around these proteins called histone proteins, which is why DNA is compact enough to fit in each cell. Because if you were to take just one strand of DNA, one person's DNA, and uncoil it and stretch it on a table, it'd be about 1 meter long. So obviously that's not the form that we want to store it inside of our cells. So we coil it up, and then when we need to, uh, for mitosis to happen, the DNA unravels, and it is going to super condense into something called a chromosome. And I like this picture, even though it's not. Oh gosh, I like the color coded pictures. Um, okay, well this is decent, I guess. Yeah, no, that isn't good enough. I want it to be numbered. Well, not the best either. We're gonna have to stick with the tried and true here. All right, so chromosomes are pieces of DNA. Think of it like a pizza. If your pizza is DNA, slices of it are going to be chromosomes. So each of these chromosomes, when combined, they form a complete set of DNA. Or I guess another example, if you watch Power Rangers growing up, it's like the individual attack robots that always get their butts kicked. Those are like the chromosomes. But then when they combine into the Megazord, that's like the DNA. If you don't know what I'm talking about, just search for Power Rangers um, Megazord transformation on YouTube. You'll see what I'm talking about, and you'll go, "Oh, I get it now." So humans, we have 23 chromosomes, but two of each. Like think of it as you can have 23 shoes, 23 types of shoes, but you actually have 46 because you have two of each shoe. Now, each of these chromosomes, they differ in the types of genes that they have. We haven't talked about this yet, but a gene is any segment of a chromosome that contains instructions for making one observable thing. For example, there is a part of one of your chromosomes that contains the information for making hair pigments. Maybe it's on chromosome 2. Maybe on chromosome 11, there's the gene for making hemoglobin, which is in your blood. So I guess if you wanted to think of it like this, think of your DNA as a giant recipe book that contains every recipe known to man and woman. But of course, it's hard to find the recipes you want in there. So you're actually going to split that book into 23 smaller books, each one focusing on a different kind of cuisine. Like maybe book 1 will be desserts, maybe book 2 will be Italian cuisine, maybe book 3 will be types of surf and turf, maybe book 4 will be uh, fun with subway combinations. I don't know, but chromosomes are subdivided pieces of DNA. We have 23 individual chromosomes, but we have 46 total. All right, so now that you've seen that, we're going to talk about some chromosome terminology. So what I'm going to do here is I'm going to draw chromosome 1 in purple. So this is what chromosome 1 looks like before. So this is chromosome 1. And this is before S phase. So when I teach mitosis, we basically just go very simple. I usually only use an example of mitosis with chromosome 1 and chromosome 2. But this is what chromosome 1 looks like before the S phase. I generally draw them as shapes because it makes it a little bit easier to teach with shapes than it does with just lines, because all the chromosomes look like lines. So let's say that chromosome 1 is going to be a column, a rectangle. And you have 2 of chromosome 1s. And you get 1 from each parent. So we'll say mom. And this is dad. What is different about these chromosomes? We'll talk about that later, but each of these chromosomes you have, for example, chromosome 1, is chromosome 1 has the same genes, always has the same genes. So my chromosome 1 might contain genes for hair growth, hair pigment, and hair shape or follicle shape. Um, uh, but my chromosome 1 has slightly different versions of the gene than your chromosome 1. And to complicate things, these two chromosome 1s each contain different genes or different versions of the genes. So, for example, maybe the chromosome 1 from your mom might have the gene for blonde hair pigment. Maybe the chromosome 1 from your dad has the gene for brown hair pigment. The result is that you have the term is dirty blonde hair. But we'll get into that more when we get to genetics. This is just if you're curious about why you have two of the same chromosome. Just one example of it. So before the S phase, you have these two chromosomes that look just like how they did in the karyotype. But after the S phase, something is going to happen here. So this is chromosome 1 after the S phase. After the S phase. So recall that what's going to happen is that if you recall the S phase, is DNA is going to be copied, right? And what it's going to look like during mitosis is that the chromosomes are also going to be copied. And each copy is going to be attached to the original chromosome through a protein connection called a centromere. So this here is called a centromere. Now, these are identical chromosomes, as in they contain exactly the same genes, exactly the same versions of the gene. And the same thing is going to happen with chromosome 1 from the dad. These are called sister chromatids. Sister chromatids are identical chromosomes that are the result of the S phase of the cell cycle. Oh, by the way, I didn't mention this. Please note this, but interphase is all the stages that do not include mitosis. Sorry about that. So you'll see on the outline it says interphase prepares the cell for mitosis, and it's G1, S, and G2. But anyways, after the S phase of interphase, the chromosomes will be copied. Each copy will be attached to the original, and they'll be connected through a protein called the centromere. It would be like if someone were to clone you and then duct tape your clone to you. That's kind of what's going on here. All right. So now we're going to talk about something called a homologous chromosome. Homologous chromosome. And this is the same chromosome, or I don't like that. Let me change it. Chromosomes that contain the same genes but different variants. Called alleles. And an allele is a different version of a gene. So to give you an example, if we're going to go back to chromosome 1, we'll just say that chromosome 1 from your mom, I think the first one I did was purple. We'll just say that it contains genes for hair shape and hair pigment. And we'll say that chromosome 1, we'll say that one of your chromosome 1s is curly, and I'll just say brown. And let's see that chromosome one from your other parent, remember it's going to have the same exact genes because it still is chromosome 1. But instead of curly, it's going to have wavy. And instead of brown, it's going to be blonde. Now, both of your chromosome 1s, one from mom and one from dad, they both have the same genes, hair shape and hair pigment, but they have different versions of them. We call these homologous chromosomes because they have the same genes, just different versions of them. Now, on the other hand, there's another term called a non-homologous chromosome. Let me go ahead and erase this. So if a homologous chromosome is chromosomes of the same number that have the same genes, a non-homologous chromosome, there we go. A non-homologous chromosome has two chromosomes or chromosomes that contain completely different genes. So these are homologous. Well, let's say chromosome 2. Let's say that chromosome 2 contains information for, I'll say, we'll just go ahead and say that it contains information for eye color and I don't know, finger length. So chromosome 1 and chromosome 2 would be considered non-homologous because the only thing that they have in common is that they're chromosomes. But they contain different genes. Sister chromatids are identical, they're clones. And homologous chromosomes are the same genes, or chromosomes that have the same genes, just different variants of them. Non-homologous chromosomes are two completely different chromosomes. All right, so now we're going to go through the stages of mitosis. And we are going to do this model with just two chromosomes. Chromosome 1 is going to be in the shape of a column. So this is what chromosome 1 is going to look like. This is chromosome 1 from your mom's side, chromosome 1 from your dad's side. And we'll go ahead and say that chromosome 2, we're going to go ahead and draw that as a triangle. So chromosome 2, this is chromosome 2 from your mom's side. I know I'm not super good at connecting triangles, but whatever. This is chromosome 2 from your mom's side. Or dad's side, you pick whichever one you want. So what's going to happen here is that the first stage is going to be called prophase. Now let's kind of talk about what's going on with prophase. So prophase is the first stage. Now what happens with prophase is that the nucleus begins to break down. So whenever you see a cell underneath a microscope, the nucleus normally looks like a dark purple spot. You can't see anything inside it. But in prophase, which is the first stage of mitosis, the nucleus begins to break down and the chromosomes become visible. Once again, the nucleus begins to break down and the chromosomes become visible. And I'm not really writing this out because I actually wrote it on the outline. No, I was definitely on my game. You're going to see something called a mitotic spindle form. And the mitotic spindle is a protein connected to two other proteins called a microtubule. And the mitotic spindle has a very important job. It's responsible for both elongating the cell, so mitotic spindle. It's responsible for elongating the cell. And it's also responsible for moving/manipulating chromosomes. Moving, manipulating chromosomes. Chromosomes can't move. Something has to move them. Remember that proteins are the biomolecules in our body that do things. So these proteins are going to act like a fishing rod and fishing wire. It's going to connect onto the chromosomes and move them. It's also going to expand the cell to prepare it to go into two different cells. And we're going to go ahead and we're going to see our sister chromatids appearing for the first time. So we got one. Subsequent ones, I think I'm going to, well, we'll see. I'll just try and keep up the color coding as much as possible, just as long. So we have chromosome 1, chromosome 2 become visible. Now, what's going to happen is that these chromosomes are going to be manipulated by the mitotic spindle, and they're going to be moved to the middle of the cell. And this next phase is called metaphase. And in metaphase, let's go back to the cell here. In metaphase, we have the mitotic spindle is fully formed. And what it's done is it's actually moved the chromosomes into the middle of the cell, a spot called the mitotic metaphase plate. So here's the chromosomes. And keep in mind, these are sister chromatids connected. They are identical, just in case you forgot. Okay. We have our triangles. And if you look here, each of the chromosomes are going to be connected by the microtubule that is connected to the mitotic spindle. Sorry, correction. The mitotic spindle is comprised of a protein called a centrosome, along with, I guess you can call it a wire called a microtubule. You don't need to know the components of a mitotic spindle, but just in case you're curious what these lines are. But you do need to know that its purpose is to elongate the cell, which is what this very outer wire is doing. But it's also to manipulate the chromosomes. And something had to move the sister chromatids to the metaphase plate, and that was the mitotic spindle. So you see that each chromosome is going to be connected to a mitotic to a microtubule. So it kind of looks like an onion almost. So metaphase, the mitotic spindle has manipulated the chromosomes to the center of the plate. Now the next phase is going to be called anaphase. And anaphase is where you can see the sister chromatids, which will be no more sister chromatids. Instead, they will now just be called chromosomes because they're going to be separated. So you can see that the sister chromatids actually are not going to be sister chromatids anymore. So just give me a moment as I draw these. And we have the triangles. And we have some more triangles. And we have the mitotic spindle connected to each one of the chromosomes. So the mitotic spindle is responsible for pulling apart the sister chromatids and bringing along identical chromosomes now to each side of the cell. So these 4 chromosomes and these 4 chromosomes, they are completely identical. So these 4 and these 4 are identical. And this is all done through the mitotic spindle pulling apart these chromosomes. Now the next phase is going to be called telophase. And in telophase, the cell is going to prepare to make 2 new cells. So if we look in telophase, here we go, big cell, we see the nucleus is starting to reform on each side of the cell. So the 4 identical chromosomes that have been moved from one side to the other, they are going to begin now to reform. So here, and here, and here. So there's going to be a new nuclei that is going to be formed around each of the chromosomes. And then the next phase, which happens really fast, is called cytokinesis. What's going to happen is a protein wire, in this case called actin, is going to wrap around this big cell and it's going to squeeze it into 2. So let's go ahead and draw the end result. So the end result is going to be 2 newly formed cells that are genetically identical. And this is mitosis. So at this point now, you'll no longer be able to see the nucleus. So you're going to have 2 identical cells. Might be helpful if I show you how this actually looks like underneath very powerful microscopes. So let's go to my favorite website, the YouTube. And you can see that, hold on a second. So mitosis cell division. All right, so these are actual images of mitosis. I'm going to try and narrate as it's going on. So we see here, this is prophase. And if you look here, the chromosomes are moving. That's being done by the mitotic spindle. Chromosomes don't just move on their own. And you see that they're being moved into the middle of the cell here. And metaphase is one of those blink and then you'll miss it. Yeah, exactly. Here, let's go back here. So right now, this is metaphase, anaphase. So you see the chromosomes are being pulled apart. And now telophase. You see the nucleus being reformed. Okay. You can see a very similar thing here. This is going to be, okay, I guess they paused it for us. This one is my favorite one because you can really see the entire cell. So this mass here is one cell. You see that the chromosomes are being lined up in the center of the cell. That's going to be metaphase. And there's this stray chromosome here, needs to move to the center. Now, when that happened, we have anaphase. There we go. That's anaphase. And now we're going to have telophase and cytokinesis right there. Now we have 2 cells from 1. Okay, well, everyone, I hope you enjoyed this lecture. This is probably going to be one you'll have to watch more than once. You're probably probably seeing a trend here. But yes, that is your lecture on mitosis. And next week we'll be having another scintillating lecture on meiosis this time. All right, everyone, as always, reach out with any questions. Over and out. All right, everyone, this is attempt 2 of recording the meiosis lecture. I actually recorded a pretty darn good meiosis lecture until I didn't, because I didn't press the record button. The pleasures of teaching by Zoom. It would be like if I taught you in class and something happened and I don't know, we went back in time an hour and I had to teach a lesson again. That's kind of how this feels like. So yeah, let's run it. Let's run it again. All right, so we're doing meiosis, and you may recall from mitosis that the goal is to create genetically identical diploid cells. However, the goal of meiosis is to create genetically unique haploid cells. And what is a haploid cell? A haploid cell is a cell that only has one copy of each chromosome. So meiosis. The goal is to create 4 genetically unique haploid cells. And this occurs in 2 types of structures. Occurs in either the ovaries or the testes. And the process, and I don't know if I said this, but the ovaries and the testes, they create either eggs or sperm. And these structures here, they're called gametes. So eggs and sperm are called gametes. All right. Now, once again, let me go ahead and draw some chromosomes. So anyways, meiosis is separated into 2 stages. We have meiosis 1. And meiosis 1 is where genetically unique diploid cells are made. So genetically unique diploid cells. So what's going to happen here is that these cells, they also go through interphase, and they go through the S phase, and they are going to be, and when the DNA is duplicated, you're going to have sister chromatids just like the last time. And let's go ahead and let's draw this cell out here. And the first stage is going to be called prophase 1. Let me erase this here. And in prophase 1, it's important that we know the different things that happen in prophase. So prophase 1, we first see that the meiotic spindle, yes, it was called the mitotic spindle, but now that we're in meiosis, it's called the meiotic spindle. The meiotic spindle is going to form. The nucleus is going to begin to break down, and the sister chromatids will become present. Now, I'm going to draw, once again, chromosome 1 and chromosome 2. And we're going to go ahead and say that chromosome 1 is going to be squares, and chromosome 2 is going to go ahead and be columns. So we see here that if you remember, there's 2 copies of chromosome 1, 2 copies of chromosome 2, one from each parent. At the end of S phase, the chromosomes are copied, and that's when you have the sister chromatids. Wait, why am I doing it the same color? So this is just as we have normally. Now, This next stage, which is going to be meiosis or metaphase 1, this is the stage where I need you to keep very close attention to. Because what's going to happen during metaphase 1 is there's going to be a very important phase called crossing over. Now what's going to happen is that the pairs of homologous sister chromatids, they are going to form what's called a tetrad. So what's going to happen here is that they are going to stack on top of each other. And you might recall during mitosis, the sister chromatids line next to each other. Well, here they are lining on top of each other. So these structures are called tetrads. So this here is called a tetrad. Now what's going to happen here is something very unique is going to happen. Is a piece of each chromosome is going to overlap the adjacent homologous chromosome. So it's going to look like this if I'm going to draw an extension of the structure. It's going to look like this. So notice how the chromosomes are now crossing over to each other. And what's going to happen here is this is going to cause a process called recombination. So let me go ahead and show you more of a close-up view. So we see here that the sister chromatids, they're going to cross over just like this. And when we see this structure where one arm of a sister chromatid goes over the other, the structure form is called a chiasmata. And after this occurs, you're going to have something that looks like this. So if you see here, these new chromosomes are going to be mixed and matched with their adjacent homologous chromosome. So for example, chromosome 2 from your mom is going to start crossing over with chromosome 2 from your dad. And it's going to be completely different each time this process of crossing over happens. And these newly formed or these newly combined chromosomes are now called recombined. And the process is called recombination. And as I said, it's different each time. For example, maybe if it happens in one cell, it's going to look like this. Like so. But maybe if it happens in a different cell, it's going to look like this. These are just rough examples, by the way. It happens quite a bit differently in real life. Now, because the process of crossing over is different each time, every time that your parents decide to have a child, there are actually 12 trillion different combinations, 12 trillion different genetic combinations that can occur. Because of that, you literally are 1 in 12 trillion, and this allows for an incredible amount of genetic diversity to occur in humans. And genetic diversity is essential for the survival of a species, as we'll talk about in the evolution unit. So let me see if I can actually find us a video of crossing over in action. All my years teaching, I've never actually looked for one. Let me see if I can find a YouTube video. Bear with me for a moment. All right, I'll just do meiosis crossing over. I would like one in a real cell, actually. which I don't even know if there are any of a real cell. We do a cool lab actually that where you use beads and stuff, but yeah, gosh, no, there's just nothing that is real life. Which wait, no, this is mitosis. Gosh darn it, bacterial growth. I probably don't want to see that yet. Oh, well, I tried. Okay. Now, once this occurs, oh, and I don't want to mislead you, we still do have the meiotic spindle here. And yes, it has attached to the sister chromatids because it has to manipulate them. Now, the next stage that's going to happen is called anaphase 1. And in this one here, the pairs of recombined chromatids, the sister chromatids are going to be separated. So the tetrad itself is going to be separated. Apologies. And the newly recombined chromosomes are going to be pulled to each side of the cell. Now, in mitosis, the chromosomes on each cell or each side of the cell are identical. But in meiosis, thanks to crossing over, the chromosomes pulled to each side of the cell are actually quite different. Don't worry, I will recombine them in just a minute. Recombined. Yeah, so to give you an example of how crossing over occurs, it would be like if you and your sibling, let's say you go to your sibling and you say, hey, you know what, I actually want your head and your pinky and maybe your small intestines. So you just go up to them and start to swap things. And then maybe maybe the next day, if you were to hit the reset button, you might go, do you know what, I'll go ahead and take your left lung. I'll go ahead and take your wrist. And do you know what? I've always been jealous of your Achilles tendon. So each time recombination occurs, it's different. It's random. And we see here that the meiotic spindle is going to be the one responsible for pulling these sister chromatids. And in the interest of time, and so I don't have to keep on drawing recombined chromatids all the time, I'm going to just modify this cell here. So now I'm going to draw you telophase 1. Telophase 1. And let's go ahead. And if you remember telophase 1, or telophase of mitosis, the meiotic spindle is going to break down. You're going to have a new nucleus reform around the separated pairs of sister chromatids. And you are going to have cytokinesis, where the cell is going to become 2 new smaller cells. So this is going to be cytokinesis. So cytokinesis. And this is the end of meiosis 1. All right, I'm going to pause this and take just a quick break. And in just a moment, you will have the next round of meiosis. Okay, everyone. So now that we finished meiosis 1, if you look here once again, we have 2 genetically unique diploid cells. This differs from mitosis because at mitosis we have genetically identical diploid cells. So what we're going to do now is just take one of these cells and undergo meiosis 2. And I'll make my life a little easy. So we're going to erase one of these cells because the process is the same in both of these cells. So there's no point for me to do it twice. Just know that this happens twice. Okay, select. Let's move this down. Oh my goodness. No, I did not want that. All right, that's a little better. So now we're on to meiosis 2. And meiosis 2, the goal is to create genetically unique haploid cells. And the first stage we're going to have here is called prophase 2. You're going to see here there's a lot of similarities between meiosis 2 and mitosis. And we see here that the nucleus is beginning to break down. The sister chromatids that have been recombined are visible. The meiotic spindle has reappeared. And now we are going to have metaphase 2. By the way, these sister chromatids being lined up in the center actually was a complete mistake. Well, not mistake, but an unattended placement. But do you know what? It's just as perfect for metaphase 2. So now metaphase 2, I'm going to go ahead and erase this. Erase the nucleus. Is now, of course, in metaphase. The nucleus is completely eliminated, and the meiotic spindle is going to manipulate the sister chromatids into the center of the cell. Remember that the mitotic and meiotic spindle also widens the cell too. And this is going to be metaphase 2. And metaphase 2, not nearly as much is going to happen here. Metaphase 2 is very similar to metaphase of mitosis. And now we are going to have anaphase 2. And now in anaphase 2, we finally have the separation of the sister chromatids. So anaphase 2. So we have the recombined pairs of sister chromatids are now going to be pulled to the opposite side of the cell. And this is all done by the meiotic spindle. Let me recombine these now. And now, after anaphase 2, we are going to have telophase 2. And excuse me as I go ahead and do the same exact thing before. Now that the unique pairs of chromosome are on opposite sides of the cell, the nucleus is going to reform. The meiotic spindle is going to begin to break down. And this is telophase 2. And then we are going to have the cell split into 2, which is going to form cytokinesis. And at the end of cytokinesis, you're going to have 2, well, actually a total of 4, genetically unique haploid cells. Because as you see here, on each side of the cell, there's only one of each chromosome. And it's going to look like this. Let me go ahead and recombine them. I know, not the best recombining effort in the world, but you get the idea. Keep in mind there's 4 of these total, because this happens in both of the created diploid cells. All right, so this is the end of meiosis 2. Now there's a kind of disorder that can happen during anaphase 1 of meiosis 1 called a non-disjunction disorder. And a non-disjunction disorder, there's actually a correlation between age of the parents and the occurrence of non-disjunction disorders. This is the incomplete separation of sister chromatids during anaphase 1. Now, if you remember, during anaphase 1, the sister chromatids have separated. So the tetrad, the newly recombined sister chromatids have separated. So if you need a reminder, this is what should happen during anaphase 1. So we see that with chromosome 1, we see that the newly recombined sister chromatids, they've separated just like they should. But in the other chromosome pair, we're going to see something a little bit different happen. If you look here, instead of the sister chromatids being evenly separated in chromosome 2 like they were in chromosome 1, instead, one of the chromosomes actually has latched on to the other pair of sister chromatids. And instead of having one sister chromatid pulled to each side, you have a sister chromatid and the other chromosome. So now you have 3 on each side instead of just 1. Sorry, instead of 2. At the end of meiosis 2, you are going to have these outcomes. If I'm going to show you what the 4 cells will look like. 1, 2, 3, and 4. So since chromosome 1 separated evenly, just as normal, you'd have 1 of each of these chromosomes in each haploid cell, like so. Recombine them. And, however, with chromosome 2, you're going to see that 2 of these cells are actually going to be disordered. So we see that these 2 cells on the bottom that I'm highlighting now, or that I'm recombining now, these are normal haploid cells. However, wait, why did I recombine them in pink? Oh well, you get the idea. However, these 2 cells here we see are disordered. Now, this newly made haploid cell is actually missing a chromosome. And let's say that these are sperm cells. Let's say that this sperm cell fertilizes the egg. It will result in something called a monosomy, meaning that the newly created organism, well, more accurate, zygote, is going to have only 1 copy of chromosome 2. Because when 2 haploid cells fuse, you're going to have a diploid cell. That's the beauty of gametes. Since they only have 1 of each chromosome, when they combine, you are going to have a genetically unique developing life form. So if this sperm fertilizes an egg, this sperm has no copies of chromosome 2. The egg has 1 copy of chromosome 2. The resulting zygote will have only 1 copy of chromosome 2. However, if this sperm fertilizes the egg, it will create a trisomy, meaning that there will be 3 total copies of chromosome 2. And to show you some examples of a trisomy and monosomy in a karyotype, This is what it would look like at the ends of or this is what it would look like if one of those disordered haploid cells fertilizes a normal cell. So this is a monosomy karyotype. And if you look here, oh, that's YouTube. Monosomy karyotype. Here we go. And if you look here, this is the karyotype of someone that has a disorder called Turner syndrome. Normally, normal individuals, they have 2 copies of chromosome 23, just like all the other chromosomes. And they can either have an X and a Y chromosome to make them male, or an X and an X chromosome to make them female. This individual only inherited one X chromosome as a result of a non-disjunction error. So the haploid cell that had the non-disjunction disorder just happened to be the one that created the zygote, and the resulting diploid life form only has one copy of the X chromosome. This results in a disorder called Turner syndrome, where the individual is phenotypically female, but is sterile and has a host of other phenotypic effects. Now, an example of a trisomy. Oh, here's another example of a monosomy. Kind of analyze what's going on here. So in this karyotype, there's only one copy of chromosome 7, so monosomy. Chromosome 9, it looks like a piece of one of the chromosome 9s is missing, probably the result of a non-disjunction disorder, which can lead to a series of phenotypic effects. Don't know what's going on with chromosome 22. I honestly don't. Now here is an example of a trisomy. So if we look here, this is a trisomy for someone that has this is a trisomy of chromosome 18. So you see that there's 2 copies of each chromosome, but chromosome 18, there's 3 copies. This is an example of a karyotype for Down syndrome. 3 copies of chromosome 21. So having more chromosomes creates disorders. It does not increase functionality. It does not give more genes. It just causes a series of complications that unfortunately occurs in the resulting organism. Let's see. This is also, I believe, trisomy 21. There's something else that can happen called Klinefelter syndrome, which is where there's 2 copies of chromosome X and a copy of chromosome Y. And the resulting individual is going to be phenotypically male, but with some female characteristics. Usually, this is individuals that have Klinefelter syndrome. They're started on testosterone therapy, so they develop more of the male characteristics, and makes them more phenotypically male. And these individuals are also sterile. So if we look here again, we see that this non-disjunction disorder has created 2 disordered haploid cells. This one has 2 copies of chromosome 2. In our example, this haploid cell has no copies of chromosome 2. If the first cell fertilizes a normal cell, it's going to have 3 copies of chromosome 2 instead of 2. And if this haploid cell that has no copies of chromosome 2 fertilizes a normal cell, you're going to only have 1 copy of chromosome 2. and this leads to a variety of phenotypic complications in the resulting organism. All right, well, hope you enjoyed this lecture on meiosis. The first recording I did I told much better jokes. That was much funnier. May have been the funniest jokes I've told in my entire teaching career, and you all missed out on it. I'm sorry. But I hope you learn about meiosis, study well for the exam, and I'll see you all next lecture. All right, so now we're going to talk about the last part of our lecture, and this is going to be cancer and the development of it. So normally the cell cycle is tightly regulated. And we talked about at the beginning how there are proteins that are actually designed to make sure that the cell progresses through the cell cycle when it is appropriate and necessary. However, sometimes mutations happen, and the genes that are responsible for cell cycle control become mutated and become either too effective or ineffective. So cancer is interesting, and there's two kinds of cancer that we'll talk about. The first is not really a type of cancer. It is called a benign growth. Now, what happens here is that cells will overdivide in one area, as in they are dividing when they aren't supposed to at times, but they grow very slowly, and they generally do not pass a certain size. They're normally pretty small. And they normally do resemble the actual tissue that they stem from, meaning that if it's, for example, I don't know, if it is pituitary tissue, they're going to resemble pituitary cells. Or if it's epidermal tissue, it's going to resemble epidermal cells. The ones that are the biggest concern are called malignant cancer cells. These are cells that divide before they are ready. As in, they're constantly dividing even when they aren't supposed to. They just divide as fast as they can. And because of that, they look strange and misshapen. So if you look here, is that the cancer cells actually do not resemble the tissues that they stem from. These are the cells that not only look different, but their activity is highly irregular. And they just keep on dividing. But what's also scary about them, and this is just for your own knowledge, it isn't what you're going to be tested on. What's scary about it is that they actually can send out signals that calls for blood vessels to be made. And if that's the case, then these cells can grow even faster, because now with the blood supply, they have more nutrients in order to grow. And when it gets really bad is when it enters something called metastasis, where the cells will actually break off and they'll enter other parts of the body and begin growing there too. And in that case, the cancer becomes very difficult to treat because it is so prevalent throughout the body. So malignant, they are misshapen. They divide well before they're ready to, and they actually can branch off or break off and colonize other parts of the body. Meanwhile, benign, they do resemble the tissue in question, and they also grow very slowly. There's two major gene mutations that result in cancerous growth. The first is called a proto-oncogene. Normally, their activity is highly regulated. But if a mutation occurs, which could either be caused by environmental conditions or genetic conditions, is that this proto-oncogene will actually have a gain of function, meaning that it's actually going to work even better, work even faster, and it's going to improperly tell the cell to keep on dividing. So this is going to actually tell the cell, hey, keep on going, keep on going, don't stop, keep on dividing. The other gene is something called a tumor suppressor gene, and one of those major genes is called P53, or one of the major proteins. We're not going to talk about that too much in this class. That's more of a Bio 107 thing. But normally, a tumor suppressor gene, whenever a cell is displaying improper activity, or just in general, a tumor suppressor gene tells the cell to stop dividing. But if it is mutated, at times the mutation will actually cause the gene to create a non-functional product, meaning that there's now no ability to tell the cell to stop dividing. So one or both of either a mutated oncogene or mutated tumor suppressor gene can actually cause the cell to divide uncontrollably, either because in the case of the oncogene, it's being told to divide too rapidly, or in the case of the tumor suppressor gene, it's being told there's nothing to tell it to stop dividing. Okay, so that is our discussion on cancer, and we'll see you for the next one.